All right. Well, let's go ahead and get going. So, announcements. Make sure that you do quiz 6 tonight, if you haven't done it already. So it's due by 11:59 p.m. tonight. I'm probably going to hand out problem set 2 on Friday. So, and we then also, in terms of group work, that group work probably will take place on Monday. Okay, so I don't think that we'll get through the material far enough to have it on Friday. So I'm pretty sure it's going to be on Monday. Okay, if I don't say anything else today, then know this on Monday. Okay, all right, and I'll put problem set 2 on Canvas. Okay, all right. So 3 lives have I, gentle enough to soothe the skin, light enough to caress the sky, hard enough to crack rocks. What is it? Water. Oh gosh, he is in unison. You're all talking to each other. Easy one. Okay, water. You're right. Okay, very good. Very good. All right. So last I guess it was Monday. We were here looking at, or finishing right here, looking at regulation of PFK1 in the skeletal muscle. So we spent most of our time on Monday going over some of the complexities of the regulation of, in particular, PFK1 in different tissue types. And so I highlighted the fact that in the liver, cardiac muscle, and skeletal muscle, in all 3 of those, that enzyme, PFK1, can be regulated by fructose 2,6-bisphosphate. However, the production and the breakdown of fructose 2,6-bisphosphate is different in all tissues, despite the fact that the name of the enzyme that does the production and breakdown is the same in all 3 tissues. Okay, but it's different isozymes and it's regulated in a much different capacity. Okay, so that's what we spent our time on Monday looking at. Now, I would also just like to highlight for you that in the liver, I extended beyond just PFK1. I talked a little bit about FBPase1 as well in its regulation. We looked at glucagon and insulin signaling, and I even mentioned PepCK, glucose 6-phosphatase, and then pyruvate kinase. Pyruvate kinase is covalently modified. PepCK and glucose 6-phosphatase are regulated through genetic expression, though we didn't go down the rabbit hole of seeing how that happens. We just said that it is. When we get into skeletal muscle and cardiac muscle, we really just focused on PFK1. Okay, what regulation is done at PFK1. F26BP, of course, I highlighted, and then took a step into discussing ATP and its regulatory capacity with regards to PFK1. It turns out that ATP, let's go back a couple slides here. I could even stop right there, but let's just go one more here. Looking at these things here, ATP is an allosteric inhibitor of PFK1. AMP, I didn't really mention this, but AMP and ADP are allosteric activators of the same enzyme. Okay, AMP and ADP concentrations will increase as, of course, ATP hydrolysis is occurring at more rapid rates, which would be expected to occur at like sustained muscle contraction, something to that effect. And we'd expect then, of course, the glycolytic rate to increase in those conditions. Okay, so that's a review, a lot of review of what we talked about on Monday. What I want to go ahead and do for the remainder of Lecture 5 is focus on something that I would call just special topics. I think that we do need to pay attention a little bit more to PFK1. Oh yeah, I'm sorry. Question. Yeah, so the question is the ATP, the ADP and AMP, are they binding in the same spot as the ATP on PFK1? Yes, they are. The regulatory sites are bound either by ATP or ADP and AMP. Yep. So they can occupy the same spot. Okay. So going back to what I was saying, today I want to spend a little bit of time talking about like special topics. I think that we'll continue to focus on PFK1. We'll extend a little bit beyond though PFK1 activity in these cells, and we'll also talk a little bit about FBPase1 as well. But I think that it's always nice to try and address and apply some of the material that you guys are learning, because the applications can sometimes be very helpful in helping you remember some of these details, and of course important as you move on to metabolism. Okay, so we're going to look at, first of all, just exercise, and we'll think about skeletal muscle, though much of what we could say would apply to cardiac muscle as well. We won't get to this today probably, but we'll also talk a little bit about type 2 diabetes or diabetes in general. And we'll apply some of those concepts that we learned from insulin and glucagon signaling to that as well. Okay, but let's think about this, exercise and skeletal muscle, and what would happen in terms of metabolism in the skeletal muscle. Okay, and there's some things that we can highlight here that would be more general principles as well. All right, but let's start just big picture again, looking at this figure here. This figure is not from your book, it's actually from a different book, but that figure is going to represent essentially where energy is going to come from to sustain muscle contraction. So if you've looked at the contractile units of muscle and looked at the mechanism of that, ATP is hydrolyzed ADP in that process, okay? And ADP then would be released by the muscle contractile unit and then hopefully converted back to ATP. Right, so if you were to go from rest, so right now you are all resting, I'm resting. If we were to all of a sudden take off on a sprint, this figure here over to the left demonstrates where the energy would come from to carry out muscle contraction. Right, all of us have in our muscle fibers and in our muscle cells a certain steady state level of ATP. Okay, and for the first couple of seconds, and there's a really short period of time, that ATP would sustain muscle contraction, meaning that you would have a supply there to run for about a second at a full dead-on sprint, a second or two. Right? Some of you think to yourself, that will be enough for me. A second or two of a full dead-on sprint, I don't need to run anymore, and so I'm just going to stop right there. Some of you are a little bit more serious about this kind of thing, though, and so you would want to keep going. All right, but what we see here is this precipitous drop in ATP in terms of what is supplying the energy for the muscle contraction, okay? All right. There has to be something to then replace that ATP. It's not like ATP is going to run out, and so there's, this graph is not great in that sense. ATP won't run out, but the amount of ATP that you have in your cell would last a very short period of time, okay? What will occur almost instantaneously upon sprinting, okay, sustained muscle contraction will be that creatine phosphate, then the use of something called creatine phosphate will go up. Okay, and what we have over here is essentially showing this. Now, this says phosphocreatine, this says creatine phosphate, same thing. Okay, phosphocreatine, creatine phosphate. All right, but what we see here is this. You start to take off on a sprint, ADP concentrations are going to go up. Okay, and if ADP concentrations are going to go up, there is this reaction that's catalyzed by an enzyme called creatine kinase. Okay, and creatine kinase is an enzyme that's found in your skeletal muscle. It holds the reaction that you are looking at in equilibrium or at equilibrium. So it's a reversible enzyme. So if ATP concentrations go up, that will cause this to shift in this direction, and this molecule phosphocreatine will donate a phosphate group to ADP, making ATP. Okay, so ATP concentrations will go up. Essentially what we're saying here is that creatine phosphate or phosphocreatine is a supply of ATP in short bursts of muscle contraction. Okay, certainly just endogenous ATP, whatever's there, and then followed up right by phosphocreatine. That will start to supply the needed ATP to continue to sprint. Okay. Now again, you don't have much creatine phosphate, right? And it will start to drop off in terms of being able to produce ATP. You don't have that much of it. We're talking it's going to drop off in second timescales. Right? So during that period of time, what will start to happen, as we see here, is that some form of metabolism, it says aerobic or anaerobic, really it's anaerobic metabolism that I'm going to focus on. Some form of metabolism is going to kick in and you're going to start to take carbon sources and start to generate ATP from those. Okay. Carbon source, of course, was going to be glucose in this case, rapid supply of energy to the cell. Glycolysis is the pathway that, of course, is going to be doing this. Okay. So what will happen in that short period of time, within seconds, it's very important that glycolysis, the rate of glycolysis, starts to increase. Okay. Now this graph is not great because it has it going all the way into hours. None of you are going to sprint for an hour. I don't care how serious you are. Okay. You're going to fall down before that. Okay. But certainly the glycolytic rate is going to respond in very rapid fashion and start to increase ATP production. Okay. Now what we want to do is look at how that's going to be the case, and also think a little bit about this in terms of quantitatively what needs to happen as well. Okay, question, yeah.
So is this graph just showing like strenuous exercise, like sprinting, and not like longer running, for example, like
yeah.
for an hour?
Yeah, okay, so the question is this graph just showing strenuous exercise, or could it be also applied to or applicable to, a marathon or something like that, a much longer run? It could be because you have aerobic metabolism going on here. I'm not sure why it starts in the minute time scale. I haven't really investigated that. This is obviously stylized data. And I'm not sure where they took this from, a primary journal article. But you could apply it there. I will say this, that carbohydrate metabolism certainly is something that would be relied upon in a run like that. But fatty acid metabolism will become super important in that as well. And fatty acids can only be metabolized in an aerobic environment. Okay, so you don't metabolize, at least not from an energy standpoint. You can't get much energy from them without oxygen. Okay, one of the things that I don't know if I've said this or not, glycolysis is one of the few pathways that can actually produce ATP directly. Okay, independently of oxidative phosphorylation. Okay, because it has two steps where it will carry out a substrate level phosphorylation. Right? Fatty acid metabolism or lipid metabolism, there is no substrate level phosphorylation in that pathway at all. Okay, so it relies on breaking down lipids, feeding them into the citric acid cycle and oxidative phosphorylation. So you'd have to have oxygen there. Okay, but this could be, I think, applied to that as well because they do have the aerobic metabolism, which is what you'll be looking at. Okay. Other questions? Considering that question, we'll be considering only really strenuous, take off on a sprint, that kind of thing, at least for the time being. Okay. All right, now let's do this. Okay, let's move forward, and let me go ahead and ask you a question to kind of set the stage for this quantitatively with this. Okay, I'm going to go ahead and expand this. Pause that. Okay, so the question says this. It's kind of a complicated question in terms of the length of it, but it says ATP production through lactate fermentation, that's something that would be, of course, occurring during our sprint, is only 1/16 as efficient as oxidative phosphorylation, about 1/16 of efficient. If glycolysis flux increases 9-fold, so there's going to be an increase in the rate of glycolysis, while the cell loses the ability to carry out oxidative phosphorylation, which of the following will be true? So we've got a pathway that says 1/16 as efficient, but it's going to increase 9-fold. Which of the following is true based off of those factors? Okay, ATP producing capacity from glucose has increased 16-fold in the cell. ATP producing capacity from glucose has increased 9-fold. ATP producing capacity has decreased 2-fold or has decreased 16-fold. What do you think? So talk to your neighbor about that. Why is that the case? Why is it important? Do I really want to take off on a sprint? Those kind of things. Okay, your marks get set, go. All right. Minute or 2 minutes. I'm going to give you 2 minutes to discuss that. Okay, about 10 seconds left, so submit an answer if you haven't done so already. Okay, 3 seconds. 1 second, all right. Here we go, let's check this out. Now I'd lock, there we go, show results. Okay, 67% of you think that ATP producing capacity has decreased twofold in the cell. So let's go ahead and look at this. Let me set the stage for us here. With this, come back here. So we've done this. We've looked at something like this where we take glucose, break it down to 2-pyruvate. Okay, ATP, of course, is going to be produced. So you have the ATP, and they have to use the NAD plus. Right? So we have 2 NAD plus, convert it to 2 NADH, and then we have the 2 ATP. And I'm just going to write net results here. Okay. So we've got that pathway. Now, we could draw below that pathway, and we'll get into these things as the semester progresses and continues to progress. But pyruvate can get in here, and we won't draw anything really specific here, but the citric acid cycle and connected to oxidative phosphorylation, those processes are going to lead to an additional 30 ATP. Okay. Mechanisms forthcoming. We'll talk about that later. All right, but what we have then, right, in terms of in an aerobic environment, because this requires oxygen needed, okay, make 32 ATP. And I've said before, if we lose oxygen, then we essentially lose this capacity, right? These go away, and that 30 ATP goes away with it, which means that we're left with a pathway that can make ATP that is 1/16 as efficient as the full scale, let's convert all of glucose and the carbons in glucose to carbon dioxide and water, okay? Now, I then presented this case, okay, so we lose this is gone. Let's just say that the glycolytic rate increases 9-fold. Okay, well, if it increases 9-fold, which is a pretty significant amount, right, we can take that pathway that's 1/16 as efficient, multiply it by 9-fold increase, and that's going to be equal to 0.56-fold increase in ATP producing capacity, which is a 2-fold decrease. Right? So even increasing the glycolytic rate 9-fold is not going to cut it. You're not going to get as much ATP. You're going to get half the ATP that you would normally need to sustain cellular activity, and of course you're using more in a sprint. Now, I bring that up for good reason. So, by the way, 67% of you were correct. Good job. Okay, so good job. It would be a 2-fold decrease in ATP producing capacity when we look at this. About 9/16 as efficient. Okay, even with an increased rate in glycolysis. Now, let's think about this then from an enzymatic standpoint. Okay, we've been talking about PFK1 for the last couple of days. If you think about that enzyme, let's say that at rest, what you guys are doing right now, let's say that enzyme is at 10% of its maximal velocity. Okay, maybe it's a little lowballing it, but let's just lowball it. Okay, let's say it's about 10%. That's probably be a little bit higher than that just to sustain activity. Right? Let's say then it jumps up to 90-fold, or 90. I'm sorry, not 90-fold, but 90% of its maximal velocity. If it were to do that, then that would be a 9-fold increase. That's a huge increase in regards to going from 10% to 90%, right? If you come back to this graph, I overshot it, but if you come back to this graph, right, going from high ATP to low ATP, right, and we have steady state fructose 6 phosphate concentration, I have it going here from 25% to 90%. That's not a 9-fold increase at all, okay? You'd have to go even lower than that. That's a big increase in enzymatic rate, okay? But not even close to what's needed if you were to take off on a sprint. We need something more like, come back here, we need something more like this, an order of magnitude larger, and 90-fold. Because if we get something 90-fold, then that will be a 5.6% or fold increase in ATP producing capacity, and now we're getting 5 times the amount of DNA out of glycolysis that we would normally, which is needed because you're sprinting. Okay. So I paint that picture to try and say, okay, what's going on? PFK1 is, right, even if we go from 10% to 90% of this maximum velocity, is not going to cut it. Right, so what else is going on here? Okay, and that's going to bring up something else, something called what's called a substrate cycle. Okay, so let me ask you another question. Okay, this is a yes/no type question, but think about why you're saying yes or why you're saying no. I might ask for some brave people to explain this to me. All right, and there's no real right answer to this because I'm asking you, would it be expected? All right, so would it be expected that FBPase1 would be expressed in the skeletal muscle? What do you think? Do you think that enzyme is in the skeletal muscle? And if so, why do you think it's in there? Or is it not there? And why is it not there? Okay, so let me give you a minute 45 to answer that question. Your marks get set, go. What do you think? Yes or no? If you're wondering, FBPase1 is catalyzed as one of the last steps in gluconeogenesis. The dephosphorylation of fructose 1,6-bisphosphate to fructose 6-phosphate. Okay? About 15 seconds left, so submit an answer. All right, 5 seconds. Okay, let's check this out and see what people say here. All right. Close to being a 50-50 mix here. Okay, so 53% of you say no. Is there someone brave enough to say why they say no? Yeah.
It's like kind of a shot there, but
Oh.
so the cell or the muscle cells aren't going to be able to tell if there's low glucose in the blood because they don't have the ability to sense it like the pancreas.
Okay.
Because of that, they're never going to be like, oh, there's low blood glucose. I need to release glucose.
Okay. All right. So you're going from a standpoint of they're not really sensitive detectors of blood glucose, and so there's no real reason for them to like produce glucose and then release it. Okay. Anyone else? Yeah.
Well, gluconeogenesis takes a lot of ATP equivalents to remake glucose, so I feel like it'd be inefficient for a muscle cell that's trying to use glucose to make energy, to use energy to remake glucose.
Okay. Yeah. So yours is from an energy standpoint. It doesn't make sense, energetically speaking, for it to make glucose. It should just absorb it from the blood. Okay. What about yes? Anyone want to say a yes answer? Yeah.
So muscles can only pull in glucose like at certain points, like during insulin sensitivity
Okay.
and stuff. So when you're doing exercise, it needs to get its energy from somewhere and its carbon source from somewhere. And so if it runs out of glucose, it needs to have something else to like build back up. So what we're going to talk about makes glucose. Maybe there's some other like long-term glycogen or something that it can make to store the energy when it has high ATP levels to use it later.
Right. Okay. So your thought is, okay, well, it seems that, if insulin's, the only way that glucose gets into the blood or into the muscles is if insulin's present. Okay. So what if you go through this significant source of exercise, you run out of glucose, there's going to have to be some source of replenishing that, right? And possibly FBPase1 would be part of that pathway to do that. Okay. Interesting. Okay, so let me say this, okay, because this is the question is just what is your expectation based off of what you learned, and so first of all, FBPase1 is found in the skeletal muscle, okay, but Pepsi K, pyruvate carboxylase, and glucose 6-phosphatase are not, which means that there is nothing else to make glucose. So FBPase1 is one of 4 enzymes needed to make glucose through gluconeogenic pathway, but only 25% of those enzymes are found in the skeletal muscle. So it can't actually make the glucose. Okay. To go to your point, it actually stores a major amount of it in glycogen. Okay, and it will stop. All things will stop in terms of anaerobic before it actually runs out of glycogen or glucose. Okay, so or it will start to resort to other forms of like fatty acid oxidation, that kind of thing. Okay, so the enzyme FBPase1 is there even though gluconeogenic capacity is not there. Okay, which is an interesting aspect of skeletal muscle physiology, and also an interesting question for biochemists as they look at that and say, well, why have an enzyme that's part of a pathway in a tissue that pathway is not expressed fully? Okay, so what's the purpose of that? So let's go to this, right? And this is a slide that illustrates here PFK1. On our left, we've been talking about PFK1. We've been talking about its regulation for quite some time. And then we have FBPase1, okay? And we're going to put this in the context of skeletal muscle, except for in skeletal muscle, this is not going to happen, okay? And you could think about the rest of the gluconeogenic pathway coming up in this way, and it's not going to happen either. So this is just kind of the loner sitting there. And what's the purpose of this? And there's a very good reason that I'm bringing this up. And I'm going to try and illustrate that and make the connection back to our required need of fold increase of glycolysis in terms of rate. Okay, but FBPase1 is there. Interestingly, it's reciprocally regulated by AMP. Okay, so AMP will, in the skeletal muscle, will actually reduce FBPase1 activity. AMP, as you illustrated or as I've illustrated and talked about, will activate PFK1. Okay. Now, let me go ahead. This slide is a little messy, so let me go ahead and rewrite that out. Okay. And let's go fructose 6-phosphate goes to fructose 1,6-bisphosphate. PFK1's our needed enzyme to do that. And that's part of the glycolytic pathway. Okay. And the other step that we're talking about here would be FBPase1. Okay, breaking off that phosphate that was put on up here. Okay. Now we're going to get a little quantitative here. We're going to use some numbers that are going to make our mathematics a little bit easier. Okay, but here's what we can say about this process. We can say that PFK1 activity is 10 times greater than FBPase1 activity at maximal velocities. Okay? In other words, the expression of FBPase1 is quite a bit lower than the expression of PFK1. Okay, you have a lot of PFK1 in these cells. You don't have as much of FBPase1, and because of that, then the activity of PFK1, if it's fully tilted out, will be 10 times what you would find with FBPase1 if it was fully tilted out. Okay, so we can make our job a little bit easier by just illustrating this. We can say, okay, then the Vmax, let's just say, hypothetically, these are not actual numbers, but hypothetically, let's just say that Vmax is 100 for PFK1. That's what it can obtain. FBPase1, Vmax, based off of what I just said, then its Vmax would only be 10. Okay? All right. And you say, awesome. This is so great. Okay. Glad you taught me that. Now, let me say one thing, okay, that's very important. Nolan Arenado was signed to an 8-year contract last night for the Colorado Rockies. That's really important. Did you write that down? Okay. It's good news, right? That's good news. I had to take a break from it. It's on my mind. He's got a 3-year option, which means that he could ditch us in 3 years from now, but at least we've got him for 3 years. Okay, so that's good. Some of you are like, who is Nolan Arenado and why is he talking about him? Just don't worry about it. Okay. And if you're like my wife, so I said that I brought this up to my wife last night. I said, I've got good news, babe. And she said, really? And she got all excited, like, oh, right, good news. I like good news. And I was like, well, don't get too excited. And she's like, oh, great. And she looked at me because she knows me so well. She's like, is this one of those like good newses, like you are tell me that and you get me all excited and then you tell me some stupid baseball fact? So anyway, but I told her anyway. So I worked through the relational dynamics and pressed on. So she was excited, not for the same reasons that I was excited, but I mean, she liked Nolan Arenado, again, not for the same reasons I do. But anyway, so, so what was I saying? Oh, yeah, that's right, okay. Baseball's really important. It started. I don't know if you know that, but it started. It's there, it's present, it's coming. Okay, just get prepared. I've got my fantasy draft on the 17th of March. Very important date, okay, in my life. All right, now, okay, so Vmax. Let's use these numbers. Vmax, 110. You understand where I got those numbers? Okay, hypothetical numbers. Now, who cares about all this? Well, let's look at this, okay? And there's an important point that I want to make, okay? So we're going to look at the flux through this reaction with and without FBPase 1 and make a really important point with this, okay? So low AMP. So let's draw out this low AMP concentrations. We are at rest. Okay? And we have this reaction, fructose 6-phosphate goes to fructose 1,6-bisphosphate. That's the PFK1 activity. Okay? Now, at low AMP concentrations, this will be at essentially 10% of its Vmax. Okay? AMP is an allosteric activator of this. Low AMP means high ATP, which means that PFK1 is being inhibited. You get about 10% of its Vmax, which means that the reaction, okay, PFK1's reaction, its forward rate, VF, will be essentially, if we take our Vmax as 100, which I said on the last slide, 10% of 100 is equal to 10. Okay, so we're going to say VF of this is 10. Forward rate of this particular reaction, 10. Okay. Now let's go over here to high AMP. Muscle is not at rest, right? Exercise. Okay, which some baseball players do. Okay. Fructose 6-phosphate, go back to our PFK1 reaction, goes to fructose 1,6-bisphosphate, keeping glycolysis going, PFK1 again. Okay. This is going to be at high AMP concentration, is going to be allosterically activated. This is going to be somewhere around, let's say, 90%, which is about right. Okay. So that's a big increase. 90% of his Vmax, which means that the forward rate of this, VF, is equal to 90. Right? Our Vmax, as I said on our last slide, is 100. 90% of 100, of course, is 90. And you think, this is awesome. I can't wait till you get to your point. Okay, I'm going to get to it. Don't worry. Now, let's think about this. Flux, J. Remember this term J, flux. We're going to look at the flux of this step, the rate of this step. Flux is always going to be equal to, of a particular step or of a particular pathway, the rate or the velocity of the forward minus the velocity of the reverse. Okay? It turns out PFK1 is essentially an irreversible reaction. The reverse reaction is negligible relative to the forward reaction. Okay? So we could say this then, using these numbers here. Flux at low AMP would be 10 minus 0 because there is no reverse flux. Or if there is, it's so negligible that we're not going to take it into account. Okay? So that means that flux at low AMP at rest through this step would be essentially 10. Okay? Now, let's go here. Go to this. If you're to take off on a sprint, you're exercising, AMP concentrations skyrocket, forward rate of PFK1 increases 9-fold. Okay, we've got it goes from 10% to 90%. We've got a 90 minus 0. Okay, and now flux is 90. Okay, versus 10. So the difference here is that insufficient 9-fold activity. Right. Go back to my question. At a 9-fold increase in glycolysis, you're going to get 2-fold decrease in ATP producing capacity. So this doesn't cut it. Okay, does everyone understand where I went with that? How we got all that? Some people are like, yeah. Some people are like, eh. Nolan Arenado is exciting. He's getting paid more than I am. Okay, anyway. $260 million over 8 years. That's in some people's minds, immoral. But anyway, of course, I go to baseball games, so and buy $20 hot dogs. So I wonder where that money goes. Anyway, where was I? Oh, yes. So let's go back to this. So FBPase1, trying to connect all these things. Why is FBPase1 there? Well, let's look at this. Okay, so let's bring this whole thing into focus. Okay, so we're going to do the reverse reaction. We're going to add the reverse reaction by having FBPase1 present. Okay, FBPase1 catalyzes that reverse reaction. Now, at rest, remember, go back 2 slides. AMP is an inhibitor of FBPase1, which is AMP is at real low concentration, so we're not inhibiting FBPase1. FBPase1 activity will be about 90% of its maximal velocity, and 90% of Vmax. Now, the Vmax for FBPase1, as I said, is about 10. Okay, so this is going to be a VF. Actually, we're going to put it as VR because it's the reverse. It's going to be of 9. Okay, 90% of 10 is 9. Okay, do that same thing here, over here. FBPase1 present, accounted for. Okay, 10%. It's going to be about 10% of its Vmax because now it's being inhibited by AMP. So it's going to go down, and that's going to produce a VR of about 1.0. Now you say, okay, I think I'm following you. This is fun with numbers. Okay, let's come over here then, and let's look at these J's then. Okay, so these J's now, we're going to look at this scenario over here on the left. The J of the forward at rest, forward J is 10, reverse J, or reverse reaction is 9. Which means that our J, or our flux, through this step is 1. Because we're recycling much of the F16BP back to fructose 6-phosphate at rest. Okay, using FBPase1. Now come over to this side of the equation here. We go from J, we go to our J, we have our forward rate now 90, because you're running a race now, or something to that effect, running to first base. Okay, right? 90, and now our reverse is 1, which means our J is what? 89. The difference between these two is an 89-fold difference. So by adding, here's the key material point that I'm trying to make. By adding FBPase1 into this, we produce what's called a substrate cycle. And in that substrate cycle, we can go from, a very small flux, a 1, to an 89-fold flux, which is an 89-fold increase. Remember I said we need about 90-fold increase, okay, when we go from rest to running. Okay. So by having FBPase1, the ATP producing capacity of the cell actually can have these enormous jumps. Okay. relative to if you didn't have FBPS1. Now, there are other factors to this, but this is, I think, a very important point. And I said this term, I said substrate cycles. These substrate cycles are actually pretty common in the cell. We will find these irreversible steps have an opposing reaction that's also irreversible to make this cycle and make these steady state concentrations and have much larger increases in rates through certain pathways because of the substrate cycles. Questions? No? Okay. So that answers this question. Well, why would FBPase1 be there, but the other 75% of the gluconeogenic pathway is gone? Okay. That's it. Now, that's fun, okay, and that's great. There are other things going on, so let me do this. We'll kind of close out our discussion on skeletal muscle with this slide right here, okay? So we have focused almost exclusively on PFK1. We just added FBPase1 into the mix. We've looked at it, ATP can do. Okay, so that's what we've done. We have mentioned here, so we're at rest here. We're during exercise over here on the left side of this, so you can cut this in half. Right, at rest, ATP concentrations are large. That's going to feed back and inhibit this. Okay, mentioned that. At exercise, when things are going really, really fast, right, AMP is going to activate PFK1. FBPase1 is implied in this as well. So we've looked at that, but now let's look at the rest of everything else. Because the glycolytic pathway is more than PFK1, especially from a regulatory standpoint. There are 3 regulatory enzymes, not 1. Okay? So let's look at this and let's paint this scenario. Right? ATP concentrations also will inhibit the last step in the pathway, pyruvate kinase, reducing that rate even further. Okay? If these two are being inhibited, pyruvate kinase and PFK1 by ATP, they're being reduced in rate, this molecule here, glucose 6-phosphate, will increase in concentration. You're not using as much of it. That will feed back, especially if you're sucking in glucose from the blood, that will feed back and inhibit hexokinase. Okay? Which means trapping reaction will go down. Right? And what will happen here, and we're going to get to this later on, because we're going to talk about glycogen metabolism next week, but glucose 6-phosphate will actually be favored and converted to glycogen. Okay. Right? So this whole thing, this will reverse in this direction and go that direction. Okay. All because ATP concentrations are at normal levels. Now, take off on a sprint, go over to the right side of this. You take off on a sprint, and what's going to happen? Well, PFK1 activity is going to go up. Now, if PFK1 activity is going to go up, it's going to make more fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is an allosteric activator of pyruvate kinase. It will undergo what's called feed-forward stimulation or feed-forward activation, so that more ATP would be produced down at the bottom. Okay, more pyruvate is going to be made. At the same time, then, that's going to start to drain glucose 6-phosphate, which means hexokinase will start to lose its inhibition. And we'll get to this later on next week, glycogen will start to be broken down. So that then there will be a net increase in this direction as exercise occurs. Again, why? Because AMP concentration is up, activated PFK1, PFK1 made fructose 1,6-bisphosphate, activated pyruvate kinase, glycogen starts to be broken down because they're draining glucose 6-phosphate. And we'll look at other things that will activate glycogen breakdown later on. So ATP in a skeletal muscle is very important. And AMP is breakdown product. AMP is very important regulators of this pathway. And that's the take home. You think to yourself, why couldn't you just said that at the very beginning of the class? We could have skipped all that fun stuff that you did, and we would be done. We would have been done 40 minutes ago. Yeah, I know, because I decided not to. Okay. Questions? No? Okay, one last thing here. Okay, and then we'll finish out. Okay. The other thing here, so this is important, right? AMP concentrations, exercise, activating PFK1, the whole shebang, right? Putting all that together. The other thing, though, that's happening here is this. AMP kinase is being activated. Okay, I talked about AMP kinase. I think I talked about it on Monday. I think it was Monday. AMP kinase is AMP activated kinase. And it's activated by exercise because exercise leads to AMP. Okay? That will do the following. Okay? It will lead to these things. Okay? Skeletal muscle and the heart. It's going to lead to increased glucose uptake, increased glycolysis, and if you remain aerobic, increased fatty acid oxidation. Okay? It will shut down other pathways that are going to try and use ATP to make like proteins or to make triacylglycerides or to make glycogen. Okay? So AMP kinase is this central regulator as well. Okay? So that's also going on. Okay? Now, to activate glycolysis in the cardiac, it's going to do that through the action of PFK2, okay, which we talked about earlier. It will lead to the transport of GLUT4 vesicles to the membrane of skeletal muscles and cardiac muscles so they can start to drain glucose out of the blood. Okay. And then we haven't got to fatty acid oxidation yet, so we can't talk about what it does. So the next time you go out and exercise, think about this the entire time you're exercising. Okay, think to yourself, that's interesting. Okay, it will motivate you to go further. Okay, so let's stop right there. We'll come back. We'll talk about diabetes on Friday, okay? All right.